Thank you very much for asking a microbiologist to come along and do a talk. So yeah, I'm Mark Prescott, I'm Senior Assessment Manager at UKAS. I live just across the M62 up in the north, so I was asked to do the northern leg of this. So we're really just going to have a quick look at the new standard. Obviously, as has been mentioned now, we've got the 2022 version. We're going to have a little look at the transition process and where that is at the minute and how it's working. I'm sure many of you have had your transition assessments at this point, so you'll probably be quite familiar with a lot of it. Touch on some of the trends that we've seen so far from some of the assessments and some sort of take-home messages. The second half is going to be largely around sort of validation and verification as well, the other word that's missing there. So just as a sort of refresher, as we know, the standard has been revised in December of 2022, hence its name. All sorts of people got involved all over the world with updating the new standard, including representation from the UK as well. UKAS have some involvement in that, along with many other parties. Really major changes have been mentioned to it already today. The large bits around this sort of focus on clinical risk, particularly as we were just talking about there with regards to the uncertainty and management of patients, really sort of going beyond just doing tests because it's convenient and you like it, really why are you choosing this particular assay over others that are out there. So have a little look at that. We've got a 3-year transition process. We're sort of coming up to the end of the second year of this now. Last year was really around sort of putting the gap analysis together, putting training documents out there, training all of our assessors. I'll come to that in a second. This year is the main year for doing the actual transition assessments, and we're almost in November. I think about 80% of them have been done so far, so it is moving. It's been a lot of work. There's still a lot of work to do. I think next year is the one we're probably most worried about, because that's where we'll be getting all the remaining grants of accreditation out and tidying up anybody who still needs to have an assessment or anything. Key changes, we've touched on these already, very much patient focused now. Why is it that we're doing this? The welfare of the patients is the key thing now here. Everything is risk-based, and in theory this is good for labs because it gives you a bit of opportunity to sort of think about things, not just doing things like Stephen says, it's a box ticking exercise. It isn't. But, really you justifying what you're doing and why you're doing it, and that is one of the key things with the new standard. It does give a lot of flexibility for labs to justify different approaches. A couple of useful documents there, other ISO documents related to risk. And then the other thing, of course, is the incorporation of 22870 into the ISO standard. So that point of care standard is going to be withdrawn. Anyone who wants point of care accreditation will just do ISO 15189 now, which hopefully will encourage more to go for it. So as I say there, about 700 laboratories to get through. We've got 170 assessors. They've all had to do some training in the standard. They've all had to do a test to make sure that they're ready. You might laugh, Jane, at that. So yeah, that was all assessors had to go through this. So whatever you're feeling when you have your assessments, we've had to do it as well. We've all had to pass the test. Some of us didn't pass it the first time, but we got there in the end with it. And we got 36 months to complete all of this. The ultimate deadline there, that's the one to take note of, 6th of December 2025. That's when the 2012 version will be withdrawn completely, along with ISO 22870. Anyone who's not accredited at that point, well, you won't be until you get the 2022. So that's the date that, well, it scares us as much as it might scare labs. Very quickly with the actual process itself. So it's all in the sort of UKAS technical bulletins. These sort of came out last year. Really about completing the gap analyses, submitting that to UKAS. We look at that gap analysis before we turn up on site, so we've got a good idea where you are. And then it's really just following things up on site. We have had a few issues with some of this, which I'll touch on shortly. Findings will be raised against the transition. Those of you who had your assessments will see that you get 2 sets of findings, one for your surveillance assessment and one for the transition. Once all those are cleared, then in theory you get your grant to 2022. A couple of things just to bear in mind, wherever you are in the accreditation cycle, that will not change. So if you had your first surveillance, second surveillance, you will still be at the next assessment in the year. You're not resetting again at surveillance 1 again. So overall, it has gone reasonably well so far. Been a few issues early on. Some laboratories sort of thinking they could postpone assessment. Some labs have had genuine reasons why they can't do it. Really major changes happening. That's fair enough. A number of labs early on did want to postpone because they just weren't ready. Well, that's fine because not many people were ready at the time. But so we've had to sort of take a little bit of a line with that one. And we need to make clear that we are assessing implementation of the standard. It's not the intention to implement. There are sort of major changes with the standard. There are some minor changes and there are moderate changes, and there are some areas that have not changed at all. We are focusing on the new requirements, or clauses that are completely new in the standard, and other areas which have considered major changes. The focus is on those. I think early on in the transition we were probably a little bit struggling with that. I think some of the assessors were looking at minor gaps and things that hadn't changed at all because they felt that they should. So we've had to do sort of more sort of training, if you like, on that, so to really try and focus on these major and moderate changes, and then work towards the others on a sort of a risk-based approach. There are some minor gaps in there. We're not going to completely ignore them, but if we know you're compliant with them already, we're not going to look at them any further on the transition. We'll just say that you're already there with it. Some of the gap analyses have been coming very late, some of them incomplete. An average of 14 mandatory findings and 2 recommendations per customer. That was from sort of June, as of June 24. So some of the trends that we've seen so far. I've removed the numbers there. I think in the London presentation, John left the numbers of findings in there rather than me grading them 1 to 8, but I've took them out because I don't want to scare people when you see the total numbers in there. But those are the areas where the most non-conformities are being seen, and I'm going to touch on some of these. I'm not going to go into numbers 5 and 6 very much, but I think those are areas where the changes are really quite minor or not changed at all, but we were getting findings against them. And so that's one of the areas where we've sort of had to speak to assessors. We've discussed it between ourselves at UKAS and thought, no, we don't really need to be looking at this on the transition. We need to be focusing on these other areas. So just touching on these key ones then. So the top one, well, the top 2 really are fitting under here, but the top one for me is the key one, which some of these can be considered surprising. I mean, we've been doing IQC for many, many years, but it's still an area of difficulty. There's no particular trend within that clause 737. We've had findings in all sorts of different areas. So some of it is around lack of IQC at clinical decision levels. Some of it is not justifying why you're doing particular EQA schemes. In some cases there's only one, in some cases there's multiple. Well, why are you choosing that particular one and not the others? It might be down to method groups and that sort of thing. So we are asking questions about why are you using that EQA scheme and not another one? And lack of consideration of use for third-party controls. That one's still a sticky wicket. I'm still a bit. I don't take quite as hard line on this as some people do. It was never in 2012 that you had to use third-party controls. It always said recommended. It still only says that this should be considered. It doesn't say you have to use third-party controls. But you do need to consider it. Why are you using that control? You might be using a particular control that's closer to the cut-off level. Some people might use a manufacturer's control that's near the cut-off level, whereas the third-party one could be sky high. It's never going to fail. So you could use that as justification. But it's really having these conversations about why are you doing things, and not just doing it because you think it's right, or because it's historical. We've always done it this way. Verification as well. Again, we all know about verifications. We've been doing this for many, many years. But some key findings around lack of clinical sign-off, lack of clinical involvement. I'll go into these a little bit more in the second part of the talk. Lack of competency assessment for doing verifications. That is an area now where people need to be competent in doing a verification, as I'll explain shortly. And considering your clinical decisions. It's not just choosing an assay because you like the analyzer or whatever. It's got to be clinically relevant to what you're doing. Clause 6.2 breakdown. So again, this fits in. This is a competency area again. We're all familiar with training and competency. We've been doing it for many, many years. But there's still a sort of a lack of evidence that staff are being trained and competency assessed in areas outside of what they're routinely doing. So using your LIMS system, even things like temperature monitoring, do people even know how to use thermometers and that sort of thing? Those are areas where we're still seeing gaps in competency assessments and training records. Clause 7.8, this is the one about business continuity. Everyone's got business continuity plans. Very few people actually test them. Very few people can actually show evidence that they've tested it. That was in, this has been in 2012 standard all the way through. It sort of sits under the lab director clause at the minute though in 2012, and I think people have missed it for that reason. It's now into the body of the standard, so it's much easier to see. This is quite a variable one. You can get some sort of standard DGHs where everyone does the same thing. I also think this is particularly relevant under specialist referral labs, because who do you send the work to, you know? And so certainly in those situations, it's worth having a good think about that. Not that just that you've got it in place, but have you tested it? And then this one is a big surprise. This internal audits. We're all familiar with audits. We do bucket loads of them, but we're still getting a lot of findings from the early transition assessments because just lack of a risk-based approach to doing audit schedules. I think people still have this mentality of auditing your full schedule of accreditation once a year or every 2 years, and not sort of considering the risk-based. You know, you might introduce a new assay, and sometimes people then put it at the back of the audit schedule. Think, well, it needs to be near the front, does it not? It's new, it's more important, it's higher risk because you've not been doing it long. So really just having to think about your audit schedule and not just automatically doing everything once a year. Just a few other little bits there. I'm not going to spend a lot of time on these. Acceptance testing, quite a lot of findings on this. I'm surprised about that one. I don't really know how that's happened. I think I've seen a few findings where assessors have seen gaps in acceptance testing records and raised that finding against the transition when perhaps it should have been raised against the surveillance. I think that may be why the acceptance testing has been a key area. But certainly trend management around field safety notices, you know, really getting to grips with those, not reliant on manufacturers and suppliers telling you something's gone wrong, but actively going out and looking for these field safety notices and doing something with them, acting on them. And then the last one there about non-conforming work, incident management, again, really surprised about this one. A bit like audits, we've been doing them forever. But it's really about consideration of clinical risk. That is the new area that needs to go into incident management. This is why it's been classed as it's a moderate change. So even though we've been doing it for a long time, we do now need to consider the clinical risk of all incidents. You know, is there any impact on the results you've sent out and patient care with that? So, some take-home messages from this then. So, as I say, the transition's live, it's proceeding well. Do have a good look at the standard. Getting gap analyses done nicely. We will do it before we turn up on site and then just tidy up afterwards. But I think going forward, a lot of the assessments I think we're gradually moving away from just sitting watching people do things and witnessing things. It is about sitting, having a conversation. I've been into labs and assessed uncertainty and sat down with them for half an hour and, you know, just having a discussion about it. Do that in Stephen's lab, and you've taught them to death, I'm sure. But that's what it's about now. It is not just about going watching you load the analyzer and press the right buttons and do the right things. It's taking it a bit further now and having conversations about things. And you tell the assessors why you're doing things. 2022 gives a lot more options for doing this now and having conversations about justifying why you're doing what you're doing. I'm about halfway now, which is good. So moving on to sort of just doing a section on validation and verification sort of on its own. Now, these are some of the new clauses. They've been sort of reworded. This whole area, I think, it's changed. They're classed as sort of moderate and major changes. So this whole thing now about assuring the clinical accuracy of examinations for patient testing. So your performance specifications, it's not just about, well, we want to get 90% sensitivity and specificity. It's about why you're using this. You know, what is relevant about your organisation that you need to use this particular assay. Documenting the choice behind why you're using a particular examination. And a few examples there, excuse the formatting. So patient populations, you know, a paediatric hospital will have a different approach from ones where they've got mostly adult patients. So they might choose different methods. And it's really having to think about that. And when you're doing your verifications, writing in it, we've done this because it's based more on paediatric testing, that sort of thing. Sample types, you know, venous blood, capillary blood. Is it validated for both or one? I've seen some laboratories who might be using venous blood on a capillary assay and vice versa. if you're doing point of care testing, then a capillary one might be more relevant. So again, this goes into your justification. This is why you're choosing that assay. As I say there, is it for use in the point of care or the laboratory? And then periodic evaluation of examination methods. I mean, we've got ongoing verification and re-verification at the bottom there. I'm sure you'll have heard those words on your assessments. I don't like to use those words. Those words have never been in the standard. It's all, I call it performance review. It's really laboratories looking at your tests on a regular basis to see that they are doing what you want them to do after you've done your verification. It's not re-verification. You're not running samples through again and starting what you did. It's looking at your EQA, looking at your IQC. How often does it break down? Is everybody competent? And doing those things on a sort of frequent basis just to confirm that your assays are fit for purpose. So a few things with verification that have been sort of updated. There's a new requirement for sign-off by authorised and competent personnel. This is about reviewing the results and the specifications. Who are these authorised personnel? So this is why this has got into the new standard now, that people need to understand what a verification is and be competent to do that verification. And getting your clinical staff more involved, not just having them sign a final dotted line, but getting them involved with the clinical utility of the test. If there's a clinical reason why you're doing that test, getting them involved in it and getting them to support what you're doing. And again, specifically, the standard now says what types of records you need to include. There's not a lot of them. It's basically saying that you want the specifications to be achieved, the results that you've obtained, and then confirming if the specifications have been met. That really is all the verification is. You set out at the front what you want to do, what you want to achieve, considering the clinical utility of the test as well. Do all your testing, get all your results, and then go back to the start and see if you've met the criteria. Doesn't matter if you've not, it still might be fit for purpose, but you can still consider those discrepancies if you've not got the results that you've achieved. Don't fiddle things, change things, move things around. Just think about it, because it still might be perfectly clinically acceptable. Then the final one on sort of verification, the standard is now saying that if a validating method is revised, then you need to repeat the verification. Now, there's no sort of set way to do this, and the 2012 standard only talks about revalidation of methods. So it's not calling it re-verification here. This is repeating the verification, but this is in the situations where the validation has changed. So for example, I don't know, a reference range might change, or a sample type might change. I've seen things like lateral flow assays where they say add 3 drops, and then you get the next kit insert and it says add 4 drops. They've changed it, so you need to re-verify it at that point because it's not what it was before. I've seen many labs miss that sort of thing. So again, this, as I say, this links back to the ongoing method review as well, and being aware of when a method has changed. You know, I don't work in a lab anymore. I don't know how companies pass this information on. I've seen it in the past where they might stick a slip in a kit box and just tell you kit inserts changed. We do expect labs to sort of check the kit inserts with every sort of new lot you open to make sure there has nothing changed. So really being aware of that. Validation. Now, I've got quite a few slides on validation, even though I don't know that we do this as much as, we certainly don't do it as much as we do for verification. The standard no longer talks about non-standard methods. It used to have this thing about you need to validate non-standard methods. That is not in there anymore. It's giving some more specific. So if you're using something outside of its kit insert, you need to validate it. Third-party reagents that are not validated for a given analyzer, you need to validate that yourself. Changing measurement ranges. So there are certain situations where you need to validate it. And actually, you need to be aware of these things. So, for example, as I say, if an assay is intended for capillary blood, but you're using it for venous blood, you are going to have to validate it. And that requires a lot more work than doing a verification. Validating as needed, confirming the performance specifications. This is really just the same as a verification. A validation is just the same thing. You're going to set out what the intended use is of this particular assay, and you're going to set the performance specifications. The standard doesn't now list these performance specifications. It used to give some examples, so things like trueness, linearity. They've been taken out now, really up for organisations to work this out for themselves and what you think is important and what companies think is important as well when assays are being developed. But the relevance for the validations is going to change between different laboratories. What is suitable for your lab may not be suitable for the next lab, considering things like patient populations and the like. Laboratory to ensure the extent of validation is sufficient to ensure validity for clinical decisions. So again, This is about reviewing the validation that's been done. You know, you need to have a look at the validation documents to see what's been done with this in the first place. So when you get an assay, how did the manufacturer validate it? And then you need to look at that, and that's where you'll work out whether it's fit for your purpose. You know, are the sample types what you want? Is it intended for what you want it to do? And then you know, looking at what those validation criteria are. It's not just about performance specifications, and I'll keep saying this same point again and again. It's not just sensitivity and specificity. It's why are we using this particular assay. And in terms of sign-off or authorization, who's doing the validations, exactly the same as the verifications. People need to do validations who are competent to do a validation. They know how to do a validation. When changes are proposed, so again, just thinking about if an assay does change, if the validation changes, you need to not just have a think about what's changed in terms of sample types. How does this clinically affect you? If you find that an assay is clinically fit for your purpose, is it still clinically fit for purpose after it's been changed? If the manufacturer has done some revalidations and changed things, is it still clinically fit for purpose? And again, the same as for verification, it specifies the records that are required. But again, it's all about the performance specifications, the results you've obtained, and then the statement on validity at the end. Those are the three key things that you need in a verification or a validation. Last couple of slides now. I'm not doing too bad. It's a good job I took those few slides out, though. I would have been behind by now. So yeah, these are common pitfalls that we see. This thing about setting performance specifications to be achieved, I know I've said this about 4 or 5 times already, it is so important. If you get deviations from that initial performance criteria, doesn't mean you can't use it and you've got to throw it all away. There might be absolutely clear reasons why it's failed. Sometimes labs might compare results between 2 different analyzers and assume that if you get different results, assume that the new one's wrong for some reason. I don't know why it is. I've seen this happen, and actually not everybody sends it to a referral lab to see what they got and use them as a sort of an arbitrator, if you like. So just because you've got a result different to the previous method, it doesn't mean it's wrong. So if you don't meet that initial criteria, don't necessarily worry about it. Just think about why have we not met the criteria, and have a look into it, because it may still be perfectly fine. About the same method on a new analyzer or a completely new method. Now, we mentioned ELISA. I don't know how common ELISAs are in what you do, but you may have an ELISA method. You may be using a particular kit that you've done by manual ELISA for many years, but you decide to get an automated ELISA. It might be the exact same kit. You're just putting it on a machine rather than doing it by hand. And then you need to just think, what sort of verification do we need to do here? You know, do we need to work out all of the sensitivity and specificity and everything else again? If the kit itself hasn't changed, it's exactly the same. It's just being done on the analyzer. So this is where it's worth just having a think. What is it that we need to do in this situation? Sometimes it may be just more about comparability. I've seen loads of verifications where you just compare, 10 positives, 10 negatives, and then you say you've got 100% sensitivity and specificity. That is not sensitivity and specificity. That means you've got 20 results which are the same. It is not sensitivity and specificity. So really, if you're in that sort of situation where your kit's not changed, but you're just changing the platform, have a think about what is it we need to do in this situation. And again, thinking about the clinical impact. And the validated use of the assay, it's people often, I see so many verifications that do not state the sample type. I see it a lot in biochemistry. There's a lot of biochemistry schedules out there that say serum, plasma, and this is something we're going to be working on next year, because a lot of them probably are not using serum or plasma. It's probably going to be serum or plasma rather than both. So, for example, something like glucose or ammonia, it may be plasma more often than it's serum. But very often you look at these verifications, then it just says blood or something like that. That's a non-conformity straight away if you're not going to put the sample type in. So having to think about it, is it serum or plasma, venous blood, capillary blood? Having to think about the sample volumes as well. And just mentioning the sensitivity and specificity before, I don't know if it was on the previous slide or not, I don't know if I can go back. Talking about analytical sensitivity and diagnostic, it's not just sensitivity and specificity, it's about considering things like interferences and cross-reactivity. That often does not get considered within verifications, or that may be more, maybe more relevant in some of the microbiology ones that I would look at. And the final point, just about multi-sites, there's a lot of multi-site organisations now More and more are coming on board, are getting sort of lots of sites bringing in new methods all at the same time. Really being very clear who has done that verification and where it's been done, because sometimes it just isn't clear. If you've got 4 or 5 labs introducing the method at the same time, sometimes it gets done in one lab. They do a massive verification in one lab and then disseminate it to the others for a mini verification. Sometimes they all do their own completely independently, but actually it can be quite difficult to work out from the verification report how it's been done. So I really just have a think about that if you are doing sort of multi-site verifications. And that's the lot.